So malaria is a disease caused by a protozoa known as Plasmodium through the bite of female Anopheles mosquito. So once the female Anopheles mosquito bites a human skin, the protozoa from the gut of the mosquito will get into the bloodstream of the bite person, and the parasite will begin to multiply himself in the liver of the person and spread into the periphery, where it's began to infect numerous number of erythrocytes. Because of the multiplication and birth of the schizones, which contain many merozoids of the parasite, the person will show symptoms like high fever, which is clinically termed as paroxysm, and the merozoids then infect other normal cells, and the parasite develop into the gametocyte stage. So then the parasite then will be this stage parasite will be gametocyte stage will be injected by another mosquito, and the vicious circle will continue. So saying this, malaria can be caused by different species of Plasmodium protozoa, which can be Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and the newly invented species of Plasmodium, which is Plasmodium knowlesi. So this parasite is an obligate intracellular parasite, which means its life is dependent on the red cell of the patient, because the parasite uses the hemoglobin of the erythrocyte as a food. That's why there are hemozoid droplets in an infected red cell. It's intracellular because it resides inside the erythrocyte or the red blood cells. So today we will focus on the diagnosis of this parasite in the laboratory. So malaria can be diagnosed in different kind of methods. The first or the most well-known and gold standard method of diagnosis of malaria in laboratory is microscopy, which use thick and thin smear of blood film, and the second one is rapid diagnostic test, which is usually known as RDT, and maybe there is a quantitative Buffy-Coat smear. Maybe we can culture the malaria parasite, and sero-diagnosis through the antigen antibody kind diagnosis, and furtherly, in the most sophisticated diagnosis of malaria can be molecular diagnosis using PCR. So, for microscopic diagnosis of malaria, we use different kind of materials. The first one is microscope, which is obviously, and second one we use microscopic slides like this, prepared with a thick and thin blood film, and we'll need Jimsa stain. After we will prepare with 1 to 10 ratio, which is 9 hand of water, distilled water actually, and 1 hand of a Jimsa stock solution. So having this, we will prepare a blood film on the slide. So a blood film can be prepared as a thick and a thin. So the first one, which is here, and this one is a thick blood film, and this is a thin blood film, which have different purpose. Actually, the thick film is the most sensitive because parasites are found concentrated on the same area, and here the thin smear is also prepared in order to get a single layer of the the parasite so that we will distinguish between the species of Plasmodium, maybe Plasmodium vivax or Plasmodium falciparum, because the two have different characteristics whenever they infect the RGBT. So we will see this on our next video, but for today we will prepare both this thick and thin blood film, and this film will be stained by a giemsa stain so that the parasite will get a color. After preparing a blood film, which is a thick and thin, this film will be stained by a giemsa stain to give a color for the parasite. Then it will be stayed for 10 minutes so that the stain will be imparted to the parasite's structure. And then we will dry the smear prepared, and after that we will put an oil immersion into the smear, and then we will observe it under a 100 times on an oil immersion microscopy. Then we will observe both the thick and the thin smear. So as I say, first, the thick smear is the most sensitive one because it has a high or concentrated amount of parasite. The thin one has important because it will give us a single layer of bacteria, the parasite, which is Plasmodium. Under microscopy, we may see different stages of the parasite, maybe the ring stage, which resembles a ring, or we will see a schizon stage, which is full of merozoites. So merozoites are those stage of parasite which affects or infect the other RBCs after schizon burst. Or maybe we will see the gametocyte or the sexual stage of the parasite. This is the gametocytes of Plasmodium falciparum. So we will see how to differentiate the two. So different stage of a parasite may be seen under the microscope.